Hello everyone, my name is Serhan Yılmaz. I'm a PhD student in computer science at Case Western Reserve University, and today I'm going to talk about our study, which is about the robust inference of Kinase activity using functional networks. So, first of all, with the recent developments in mass spectrometry, we have this rich data on phosphorylation, and when complemented by computational algorithms, these phosphorylation data allows the inference of kinase activities based on the phosphorylation of their substrates. And this is helpful because if we have a condition of interest, which could be a disease or this could be a perturbation like a drug, and if we want to investigate the effect of this condition on the signaling network, then we can do this by identifying some key kinases that are dysregulated with the help of these computational algorithms and the phosphorylation data. And this type of analysis has actually led to the development of novel kinase inhibitors for the targeted therapy of various diseases, including cancer, Alzheimer's disease, and Parkinson's disease. So these computational algorithms actually rely on two types of data. The first one is the kinase substrate annotations, which essentially provide information like this kinase phosphorylate this phosphosite. And the second one is the phosphorylation profile provided by the mass spec. And these are typically normalized with respect to a control sample and provided in the form of a log fold change. So the issue with these data is that we have an incomplete knowledge on the kinase substrate annotations, and the phosphorylation profile that we got is usually very sparse with a lot of missing data. And this causes some issues regarding the reliability of the inference. So in the literature, there are various methods for inferring kinase activity, but at their core, these are all based on the phosphorylation of known substrates. And we thought about how we could improve the reliability of these methods, and then we thought we have this information on the functional networks, and maybe by putting these inference methods, by considering these methods on the in the context of a network, we could improve their reliability. And so we hypothesized that the dysregulation of a kinase may happen as a result of a coordinated change in the signaling rather than an individual event. And if so, the phosphorylation of the sites in the functional neighborhood of a kinase, which are shown as yellow here, may be predictive of its activity. So to test this hypothesis, we focused on 4 types of functional information, which are shared kinase interactions, protein-protein interactions, structural distance evidence, and coevolution evidence. So to test the utility of these functional information, we used a curated data set on the kinase perturbation annotations, which essentially contained information like this kinase is dysregulated on this perturbation. And using this data set, we compared the kinases, dysregulated kinases with the other kinases in terms of the phosphorylation of sites in the functional neighborhood. And as a result of this analysis, we found that there is a significant difference in the dysregulated kinase for all 4 types of these functional information. And this is promising because this indicates that these functional information and the idea of functional neighborhood may be useful for improving the reliability of the kinase activity inference. So, motivated by these observations, we developed Rokai, which is a network-based framework that incorporates this knowledge. Rokai essentially applies a network propagation algorithm to create some refined phosphorylation profiles that are propagated over the network. So essentially, these refined phosphorylation profiles are an aggregate of the phosphorylation of the site itself, as well as the phosphorylation of sites in their functional neighborhood. And the framework of Rokai is flexible enough to be used together with different inference methods. So to benchmark the contribution of Rokai, we did a type of robustness analysis where we hide some portion of the kinase substrate links and then assess the accuracy of the predictions. So here we used a simple baseline method of mean substrate phosphorylation for inferring kinase activities and gradually investigated a set of different networks with Rokai. And as you can see, Rokai improves the accuracy of the inference in a robust manner regardless of the missingness and provides further improvements as more functional information is added. We also tested the utility of Rokai over a broad range of methods. So here each panel is a different inference method and the x-axis shows different values of kinase substrate missingness. The blue bars indicate the performance of the original predictions made by the inference methods, and the orange bar shows the performance of the Rokai enhanced inference methods. And as you can see, Rokai consistently enhances the inference methods. So overall, we found that the phosphorylation of sites in the functional neighborhood of a kinase are significantly predictive of its activity. And to incorporate this knowledge, we developed a network-based framework named Rokai. And we have shown that Rokai consistently enhances the accuracy of kinase activity inference methods while making them more robust to missing annotations. Finally, to provide an easy-to-use interface to users, we provide Rokai in the form of a web application, which is available at rokai.io. So, thank you.